right, guys. So welcome back to the second session. I hope you guys had some nice snacks to eat or lunch to eat very fast. So yeah, so we're gonna start now. So thanks, Miguel, for the previous lecture. I hope you guys had a kind of an idea of you know, it seems like you guys know a lot already about molecular biology, but it's always good to refresh. Sometimes I myself sometimes forget things, so it's kind of a nice thing to remember. So we're going to be talking in our next lecture about cellular scales. Why is this important? So this club is about synthetic cells and synthetic biology in general. And synthetic biology means that we can take regular life and modify it or make it from scratch, right? From zero. So that's kind of the main goal of synthetic biology. So to do that, we of course have to have a deep understanding of the cells and the physical constraints of the cell, because eventually the cell is kind of a mixture of components that behave in a physical way, right? So the cell doesn't, I mean, as far as we know, the cell doesn't violate the laws of physics, right? It follows the laws of physics. And for that purpose, it's important for us to know. But those laws of physics that applies for cells sometimes are more complex. And for that, I recommend you a book from Erwin Schrödinger, origin the What is Life. What is Life? Yes, I will send you the reference. It's a beautiful book from the point of view of a physicist of what is a cell. So let's get started. So let me just test something real quick. I want to test my pen and see if I can. Can you guys see that I'm writing?
Yeah.
Perfect. Oops. Perfect. All right. So just as a summary, Miguel was explaining this. So one of the main things that we all biologists need to understand is they call the central dogma. It sounds a bit religious, but but it's basically kind of the basis for all what we do in biology, and it's the information from the DNA up to proteins. So as we know, Miguel already explained, DNA goes to mRNA from transcription and then translation from proteins. So the proteins are basically the machineries of the cell. They have different functions. These are kind of the gadgets we will cover in our second lecture. And the kind of a nice analogy for this is if you have a message, for example, something written in Word, this is kind of your message that you store in your computer, right? It's not modified, you just have it there. Now you want to translate this message into something physical, then the computer has to read that code and translate it into a printing kind of code, right? So that the printer can read that information and make it in a physical paper that can be, distributed to people, etc. So that's kind of analogy of what the DNA versus the RNA is. And I find it kind of, an easy analogy just to keep in mind of how things work in the cell. So the RNA, as you know, as Miguel was explaining, it's kind of a more compact message. It's not as wordy as the genome, but it has all the, necessary information to produce the protein, the machines that the cell has. So, all right. So then let's move forward. So we're gonna do a little poll. So I'm gonna start with a little poll that you guys can answer um, there. So basically, what is a cell? Please go ahead and click your answers. Very good. Very good. So a lot of people are choosing basic universal light. That's good. And some other people are choosing artificial unit with some membrane cytoplasm. Very good. Very good. So this is an interesting question, because how do we know that something is alive, right? So is a cell alive, or is something that we make from scratch also alive? So that's kind of an interesting question that we're gonna try to address. So this is basically the results. So a lot of people chose unit of life, but others shows artificial unit of life. So those answers might be actually right, you know. So based on how do we define life? So let's move forward into that a little bit. So so what are the components of the cell? So can you guys tell me what are the components of the cell? What do you think are the components of a cell in general? Just write it in the chat, or you can speak up if you want. What do you guys think? So a cell needs to have, sorry?
Yeah, it needs to have something that tells it apart from its environment, and that's like
Exactly.
the layer, the what was it?
Yeah, exactly. So
Bilipid layer.
correct.
Sorry.
So a cell has to have kind of a membrane, right? So sorry, let me just annotate. So sorry, I'm just trying to use this tablet, but I think it switched to the wrong direction. Technology sometimes is a bit weird. Let me just try something. Can you guys see still my screen?
Yes, including the next slide and.
Okay, then that's not what we want. So yeah, so annotate. Yeah, it's just okay. Sorry, there's a problem here with the. I think I solved it. Okay, so yeah, so basically what we have is as you said, then we have kind of a, sorry. So we have kind of a membrane, right? Or something that isolates the cells from the environment. Some people are saying that we need the nucleus. Some people say that we need some kind of mitochondria, so something that gives energy. What else? Cytoskeleton, proteins. Exactly. So a cytoskeleton, basically, I mean, we can represent it like little lines. So those are right. Those are right suggestions. But this is what we know from the cells that we know, right? So these are the cells that we're used to see, the cells, the mammalian cells, right? So that, but if we think about it in more kind of a general way, right? So the cell has to have, first of all, as what's your name? Sorry, I forgot. We need to have a boundary, right? So it needs to encapsulate the components of the cell. So the membrane is very important. The cell needs to have some kind of energy, which is what the mitochondria does. So that's very important as well. So we need kind of a source of metabolism. But the cell also needs to have some kind of information, something that has the instructions for the cells to make another cell, right? So sorry. So if the cell needs to kind of duplicate, okay, sorry, this is just not working, but it needs basically that information. So if we think, for example, let's imagine if we are kind of in another planet, right? And for example, we have some kind of chemistry based on not water, but something else. Let's say methane, what the cars produce. But it also divides, it also grows. Can we call it life, for example, right? So these are kind of the questions that people ask themselves, but we have a very oriented way of thinking in terms of um, biological life, the life that we know. So, for example, this is kind of an animation. So, from a lab from Jack Szostak. So he's studying the origin of life. So basically, one of the theories about how life started on Earth is that it started in this kind of vents. So vents that have different temperatures gradients, and in those gradients, macromolecules, like for example, in this case, fatty acids, which are the main components of the cellular membrane, can easily be formed. And those components can then assemble just by physics, just by hydrophobic interactions. They can assemble together and start to form like little vesicles, right? So those are kind of um, one of the things that people have proposed. And that particular, let's call it protocell or first cell, um, needs to develop other mechanisms, right? So just a membrane, it's not enough, right? So it's just simply kind of isolating the components, but we discussed that the cell needs something more. So what about, for example, transport, right? So if we go, let's see what's happening. Sorry, it's just I need to unclick the annotate button, then I can. Yeah, it seems like annotation, it's not working very well. So what I was saying is the cells also need transport. So this is another animation by the same lab where they illustrate, for example, nucleotides going into the cell in this protocell. So just a simulation of showing like how in certain cases things can penetrate the membrane, but that's usually rare, right? So the membrane is actually very impermeable to many things. So one of the things that people have tried to figure out is how did the initial cells kind of solve that problem when there were no proteins? And that's kind of the research that this lab does. Another main question that we just mentioned as well is how did the cells grow, right? So one of the things that for example, people have observed is if you have a kind of this protocell or this cell that has, um, you know, just a membrane, a lipidic membrane, how do this cell grow? How do this cell becomes two? And people have shown, for example, that if you have these lipid vesicles and you add lipids in the environment just by adding them close to this vesicle, it can also, sorry, it can also I don't know why it's having problems with this. It can also basically incorporate this into the membrane and grow. But the mechanism for that, I mean, and we will see this when we talk about artificial cells, is that it needs to be regulated as well, right? So that's kind of one of the main things. So besides the DNA and the RNA and the membrane, we said that the cell also needs metabolism, right? So what we're gonna do now is we're gonna look at the molecules in the cell, and And the goal of this lecture is to try and think a bit about what are the size of these molecules and why is that important? Because, for example, you want to look at things in a microscope, it's important to know what size of things are we looking at. And this gives us an idea to think better about the biophysics of these molecules. So then we have here in this slide, we have different kinds of molecules. I think you can recognize that there are ions, which are very small. Water is one of the main components of the cell. And the phospholipids that we just saw in these animations, the sugars, the nucleotides, and also molecules that are used for energetic processes. This is important. So what are the scales of this? So if we think about kind of a measure of molecules, I think Miguel mentioned this. Small molecules like water and ions, they are around 1 nanometer, right? So this is kind of one of the things that we can have as a ruler in our mind. What about, for example, ATP? ATP, it's around 2 nanometers, for example, right? So this is what the cell uses for energy. One of the things that, for example, it's important to also keep in mind is that these molecules undergo different changes in the cell. So they undergo different reactions. So they can also become bigger by joining with other molecules. So this is one of the key properties of biological cell. So I'm gonna launch another poll. Let me see. All right, so what do you guys think? What is bigger, RNA or proteins? Ah, that's interesting. That's very interesting. Okay, I think most of you have voted. There's some people still. Okay, very good. So I think most of us have given an answer. So I'm going to end it now. So a lot of you guys said that proteins are bigger than the RNA. So I'm going to show you the answer. So actually, the RNA is bigger than the protein. So, and I'll tell you why. So if you think about the RNA, for example, in this case, this is the RNA that encodes the myoglobin protein, right? So myoglobin protein is, as you know, belongs to the muscle. So the size of that protein is around 5 nanometers. And the size of the gene or the mRNA from that gene is around 40 nanometers larger. Now, it's important to mention, as we saw before, the nucleic acids have to be compacted. So they have a lot of information. So think of this as if you have files in a computer and then you compress them to save space. The information is still there. It's just, you know, in a reduced space. So the same happens with the mRNA and the DNA. But the information that produces, like if you once you unzip the file and you printed it, it is the file, right? So it's a similar kind of analogy here. So in this case, yes, Santiago?
Sorry, what does UTR stand for, Andres?
This is an untranslated region. So this is kind of a regulatory region that it doesn't ends up being in the protein. It's just there as a regulatory region. So that's
Great.
what.
Thank you. Mhm.
So, and that's actually important because as we see later, one of the main problems that the cell has to fix is how to compact the genetic material. So that's good. Now we have another question is how big is the average protein? So these proteins here show different sizes. So one of the proteins that probably most of you know is insulin, which helps diabetic patients. The insulin is around also 5 nanometers. If we see an antibody, which is kind of produced to fight infections, it's almost 3 times the size, right? So, and if we look at a very important protein in the cell, which is the one that makes ATP, it's even larger. So the proteins, which are the machinery of the cell, are kind of, some of them are very big. And the metabolites, as we know, it's around the 1 nanometer scale. So we have kind of a 100-to-1 ratio. So sometimes the machineries are huge, right? So it's 100 times bigger than the molecule they are trying to modify. And why this is important, you will think, because the cell is going to be crowded. So if we think about a cell that has, for example, a bacteria that we will see later, like E. coli, has two micrometers, which is around 2000 nanometers, right? So the cell has to be able, I mean, in large, but the cell has to be able to store all the proteins that it needs. And some of these proteins are going to occupy a lot of space. So the environment inside the cell is going to be crowded, right? So let's look at, for example, another protein. This is just an interesting fact. The enzyme Rubisco. Do you guys know Rubisco? Rubisco is the most abundant enzyme on Earth. What does it do? It's the protein responsible in plants to fixate CO2 and make it as make it energy. And that enzyme is gigantic, right? So it's basically a protein that has different copies of itself, and it basically has kind of the ability to extract 10 times more CO2 that produced by humans. So humans, all the CO2 that we produce in the world, in the planet, this is the enzyme basically taking care of it. And it does it very efficiently. The problem is that humans, they have, we have ways of, for example, with deforestation and the usage of chemicals that prevent, that allows accumulation of CO2, even at higher levels, that these machines sometimes it gets short. Yes?
Yeah, so I had a question. Do larger proteins have a higher chance of being defective because since they're made up of way more amino acids and therefore they're made up of more, or they require more nucleotides to be synthesized. So since there's more nucleotides involved, doesn't that mean it's more susceptible to mutation?
Thank you for the question, Hirsch. It depends. It's not really the size of the protein, but the size of the gene. So you're right. So if you have longer genes, the probability to have a mistake is higher. Although, as we know, the cell has mechanisms to correct these mistakes. So at the end, the cell has very low chance on having the mistake passing to the protein. Now, that doesn't mean that it doesn't happen. It does happen, because that's why we have also, problems in the cell with some mutations. For example, like the anemia, the sickle cell anemia, for example, is a mutation in hemoglobin. Just one mutation destroys the whole protein. But those, another thing to have to keep in mind is that first is the repair machine of the DNA, but also this protein, for example, Rubisco, is made of a smaller protein, and it's just comes all together, right? So it's not just one protein produced huge, but it's just a smaller proteins that bind together. So that's kind of another thing that the cell does, is that they can make copies of the same gene. So maybe one gene is defectious, the others are okay, then it can sort of replace that, right? So that's kind of another strategy. Someone had a question.
Yes, I think it's me. I wanted to ask, when a ribosome is creating a new protein or an amino acid chain, how reliable is it? Like, is it 100% reliable that's always going to make the same one, or is it sometimes defective?
So that's a good question. So basically, that's it's actually very reliable. So the mechanism, so there's a concept in biochemistry that is called kinetic proofreading, and it's basically how proteins recognize without having without making mistakes. So, and one of the things is that, for example, things that you want to prevent mistakes, they bind very lightly. So, and for example, in the case of the ribosome, it's important that the tRNA that gets there, it's the right one, right? So, but now you have to think about also that different codons can also codify for the same amino acid, right? So, so the protein kind of also there, that's one thing. So that kind of reduces the risk of, incorporating amino acids that are not. But there's also quality control. So if a protein, for example, falls in the wrong form, is degraded in the cell. So the cell has ways to kind of control mistakes that are done at the level of translation. But so it's pretty accurate. I need to check the rates. I can tell you by DNA. We'll tell you in the following slides. But it's in the order of maybe 1,000. But I can find out the exact number for you. Brian had another mutation.
Thank you.
Yeah, Brian had a question. Okay, yeah. Okay, perfect. So as we were saying, for example, in the case of Rubisco, so we were saying that proteins can be relatively small in terms of the machineries that they belong to, like the ribosome, we saw that is huge, gigantic, but it has subunits, right? So it's codified by small little parts that get together. So that's kind of another strategy that the cell has to make. The cell doesn't have to do all kinds of proteins. It can reuse the same one protein and just make a bigger machinery. So that's one of an important concept in biology also, it's modularity, that you can reuse parts for different things. So in this case, for example, I'm showing you, if you look at the complement, which is a molecule that belongs to the immune system, it has different subunits, proteins that are very similar, and they bind together, right? So same, for example, if you look at ferritin, which stores iron, that is, important that they all come together and form a bigger structure. So that's kind of another interesting thing. Sorry, I don't know why it's not showing this, but okay, I'm gonna launch another poll. All right. So if we think about amino acids, what do you guys think is the size of a protein in average, in number of amino acids? What is the size that you think is in average for different organisms? Very good, very good. Very good, very good. right, so that's good. I'm gonna share the results. That's interesting. So a lot of people picked 300 and some other people picked 600. And that is good because you guys were right. So it's around 300 amino acids in average. So if you look at the protein length in different organisms, in E. coli, for example, in bacteria, it's around 277. In yeast, which is, la levadura, it's around 360, and in human it's around 400. So it's in average more or less around 300 amino acids. This plot here, it's basically indicating that the cells can produce more of certain proteins, right? So then you have to normalize by the abundance. So if you say, for example, proteins that are more abundant, what is the size that they get, right? So in this case, for example, the smaller proteins are more abundant in E. coli, so it's a bit smaller if you normalize by this. So, and about the question about mistakes in the DNA. So this is an interesting analogy that I found in this book. So if you look at this video, you will see that the DNA is copied in a very fast rate. So to put that in perspective, so that we know more or less to have it in our minds, what it means is that let's imagine that the DNA actually, by comparison, it's around, imagine a road that it's just 1 meter width. So it's kind of a very tiny road. And the machinery, the polymerases and all these other proteins that copy the DNA, they are the size of a FedEx truck. So it's big, right? As compared to the tiny road, as we saw, proteins tend to be a bit bigger, especially when they all get together. So this FedEx truck, which is the one that copies the DNA, so it's the analogy here, it travels at 600 kilometers per hour. So in comparison, the fastest car on Earth right now, that's what I Googled 3 days ago, it was around 435. So it travels extremely fast, right? And this makes sense because, the cells, as they're growing, it's important that they divide. It's important that they generate progeny. So this process has to happen in a fast way. Now, in the case of human, where the genome is so big, then Miguel probably is going to talk about the human genome more in detail. This process has to be more regulated. So replication of one chromosome in bacteria, let's think of it as a 400-kilometer trip. So it's like going from here, from Boston to New York. So you can find in your own countries what I don't know, what can someone tell me of a trip that took around 400 kilometers? Just as an example.
Well, from Mexico City to the nearest beach, so Veracruz, that's about 400 kilometers.
Nice, nice. Thank you. So imagine, so it's about 400 kilometers. Oh, nice. São Paulo to Curitiba. Okay, very good. So imagine that trip is replicating one chromosome in bacteria. So it's around, and the car that you're in travels like 600 kilometers per hour. So I don't know if you guys have been in a racing car. I have never been, so I haven't experienced extremely fast speed on Earth. And that trip, it's around 40 minutes, which by car is around 6 hours. I'm talking about Boston to New York. And the interesting thing is, if you think about this as replication, from every 100 trips, just one there's a mistake of one in every 100 trips, right? So, and FedEx, this company, failed 7 times every 100 trips. So the delivery, so basically the replication error, it's very, very small, right? So the cell has all these machineries that maybe we get to talk a little bit about this, that prevent mistakes in the copying of the DNA. So, all right, just by comparison, in terms of genome sizes, so we had that the genome of a virus is around 200,000 base pairs. Bacteria, as we said, is 400 million. And Drosophila is 130 million. And the human is 3.2 billion. And the plants has 150. So you can see that the plants have a ton of DNA, right? It's very common in plants that they have duplication of chromosomes. All right, so this gives us a little bit of terms of the scales in the cells, right? How does the cell handle all this information and so on and so forth? I'm gonna give you now an example in real life. So this is, for example, this is a bacteria that I looked in the lab. So I study Bacillus subtilis. It's a bacteria that is on Earth, like on the soil. You can find it pretty much everywhere. We use it as a model organism to understand bacteria and to use it for different purposes. So I'm studying this bacteria, and I'll tell you a little bit more about why I studied this in another talk. So the replication we were seeing, so imagine that this green dot here is a replication machinery. So I label DNA N, which is a gene that belongs to this replication machinery. And basically I just imaged it. So the cell divides. And there, as you can see, the replication machinery is on the center and then goes to the streams and divides in two, right? So it's basically when the genome is duplicated. So that's what happens when bacteria replicates. So if you look at the cell, it's relatively big, but the machinery is also around the size of 2 or 3 nanometers that is in charge of maintaining that replication going all the time, right? So that's how it will look in real life. So what about transcription and translation? So transcription and translation, Miguel talked a lot about this, which I don't want to get too much in detail, but one thing I wanted to highlight with the ribosome is that a lot of the mass of the ribosome is RNA. So the RNA is very important for a lot of functions in the cell, and it has, we'll discuss this, it has kind of ribozymes or enzymes based on ribosomes that do activities in the cell. So the ribosome is actually one important example. So not all the mass of all proteins is just protein. And one good example is the ribosome. So let's move now to membranes, the ones that we talked at the beginning. So membranes are very interesting, and we will talk about this in the second presentation. Membranes are around 4 nanometers thick. To have an idea what this means, one interesting comparison is an airplane, right? So the airplane has around 1 centimeter thickness of the most external shell, and the diameter can be around 5 meters. So that's about the size of the membrane with respect to the cell, right? So around 500, more or less 500 times thinner than the size of the cell. So as you can see, the shell, it's not very thick. And the membrane, actually, high percentage of the membrane is mostly proteins. So, but it doesn't mean that the membrane is less important. It's just that actually it's to say that the membrane is a very dynamic kind of part of the cell, but it's very tiny. This is an interesting experiment. So for example, they just took membranes, they make a membrane with fatty acids, and they wanted the fatty acids to follow salt, basically. So if you see, for example, here, so they create this lipid bubble, and they add some salt in yellow. And the interesting thing is that this membrane kind of it's sort of attracted to a gradient of salt. So this is a lab that works on membranes and trying to understand how membranes are kind of, working, how membranes are useful for the cell, what kind of things we can use with membranes. So this is actually, I think, a cool experiment showing what membranes can do. This is just membrane. There's nothing else inside of that bubble. So, sure. Santiago, you can.
Yeah, sorry, Andres, very quickly, I got a bit lost. Why was the membrane attracted to the gradient of salt?
So yeah, so the basically this was a simple experiment where they put the decanol, which is a fatty acid, basically, and then it forms a shell, right? And what they did is they put kind of a gradient of salt in that kind of little, it's sodium chloride in that little pad. And the idea is that the decanol kind of senses, but it's just simply electrostatic interactions with the salt, right? And it tends to migrate to where those interactions are stronger. So that's what it's trying to show here. So
okay, so
they become.
Oh, sorry.
No, go ahead.
So naturally the polarity of the molecule of sodium chloride interacts with the membrane molecules, and that kind of proposes it towards itself.
Exactly. Yeah. So this kind of electrostatic interactions is what kind of helps them, but it's just a membrane. It's nothing else, right? So
yeah,
that's what I think is interesting.
Yeah,
and,
it's amazing.
And we see, for example, what happens in cells, that cells are more sophisticated and they have proteins that helps them move specifically to different directions. So we're going to touch one last kind of part of the cell. I mean, there are different kind of parts of the cell, or not parts, but components of the cell. But let's talk about the main ones. We talk a little bit about the cytoskeleton. Cytoskeleton is super important because it helps transporting things in the cell, right? So it's kind of, you can imagine this as kind of a network of cables, right? It's kind of how people usually talk about microtubules or cytoskeleton in general. So there are two kinds of cytoskeleton proteins. So one is actin, the other one's microtubules based on tubulin. And they're very important. They do very important roles. So one is, for example, cell division, right? So if you think about, this is a mammalian cell. So the microtubules, basically what they do is they pull the chromosomes apart so that the cells can divide. So this is kind of a very fundamental function of chromosomes, but they do different things, right? So, and if you, for example, one of the interesting things about cytoskeleton proteins is that they can self-assemble on their own. So if you, for example, throw some this is actin, this is a simulation. If you have the right conditions, the right buffer, the right pH, you can, for example, assemble actin microtubules in vitro, meaning in a test tube, you take this protein, the protein will assemble on its own. And it's because of the structure of the protein and the interactions with the other subunits that allows that. And this is actually very nice because you will see later, and I will show you, cytoskeletons are really, really useful for making synthetic cells, right? Because of this property that they're sort of like a smart polymer that assembles itself. It helps a lot to carry out functions in the cell. So I will show you very cool experiments later about this. So just as a summary, this is a software that we will be working and that you will use for your project. This is basically, it's called Cell Paint. So we have, this is the spike protein of coronavirus. I just put it as an example. We have the membrane. This is the size of an actin filament, for example. We saw that they can be very long, same as actin. Proteins are about 3 to 6 nanometers, 5 nanometers in average. The ribosome is big. So it's around, I think, around 30 or so. So they can be big proteins, but it's because we have a lot of different proteins binding together. The spike protein, which is coronavirus protein, is around 20 nanometers, so it's pretty long too. So that's kind of just a summary so that you guys have an idea of the scales of the different proteins. All right, so this puts a little bit in context. So the last part of this lecture, I think I'm a little bit delayed. I'll try to speed up. So the molecules, how does it translate into cells, right? So, and I'm going to talk about something that it's very important. And as I said before, why is important to know all these sizes? Because we need tools to look at these things. So, and the tools that we use to look at things in microorganisms or in cells, because they are too small, is the microscope, right? And the microscope has limitations, the kind of how small we can see something. So then just as a summary, let's look at it again. So we have a water molecule that it's around 1 nanometer. What do you guys think is the size of a glucose molecule based on what we saw? Someone in the chat? Or you can speak up too.
10 nanometers? Maybe?
Glucose?
No. No. Like 1 nanometer, maybe?
1.5, yeah. So it's small, right? What is the resolution of electron microscope? It's around 10 nanometers. So we can see basically whatever is basically, just one sec. So whatever is here, sorry. Whatever is here, we can see it in the microscope, right? So that's kind of the beauty of electron microscopy. Now, what is the problem with electron microscopy? Someone can tell me. Does someone know? Well, electron microscopy, you need to freeze the cell basically, or the structure. You have to freeze it, not freeze it like, I mean, sure, you can freeze it like there's cryo-electron microscopy these days, which is a new technique, and Miguel mentioned it a little bit. But it also means that you have to kind of kill the cell and just make it static so that you can see it with a beam of electrons. So it doesn't allow us to see process in real time in a live cell. So that's one of the disadvantages of that. But there are some groups that are working on trying to improve this and to see in live cells as well. There's a lab near us that is trying to work on that. It's not like, sorry, that you can actually see cells live, but it's just kind of a reconstruction that they do based on just taking images of different cells at the same time. The membrane thickness, we said that is around 4 nanometers, which is equivalent to 1 centimeter shell of an airplane. A regular protein, 3 to 6. Ribosome, again, 20 to 30. What about the size of an HIV virus?
50 maybe? No.
120. Now we come to another limit, which is important. It's the limit of light microscopy resolution. So this is the limit that we use. The images that I show you of my bacteria, they were with the light microscope. So meaning that whatever is smaller than 200 nanometers, I cannot see it, right? So that's an important kind of distinction, because that's kind of a limit that it's just based on the physics of how a microscope is built. But there are some new technologies, and I will talk to you about this in a second. So the size of an E. coli bacteria is around 2 micrometers. So basically 2000 nanometers. So 10 times more than the resolution of the microscope. The yeast or levadura, it's around 6. And one of our cells could easily be 15 micrometers. This is in log scale, so as you can see, it doesn't make sense, but it's because in log scale. But now, I said that 200 nanometers is kind of a hard limit to what we can see. But there are some new technologies called super resolution, which allow us to see things that are even smaller. So we can even see viruses or things that are in that range. So for example, one technology that we use, which is SIM technology, which is super resolution, we are able to see even smaller particles. And that's very helpful to understand the biophysics and the behavior of the cell. So this is again light microscopy, resolution microscopy, and electron microscopy. Ideally, and probably one day, would be that we can have microscopes that allow us to see live cells and almost to kind of a 5 nanometer, right? So we can see basically single proteins. We can have some kind of single molecule, meaning we can observe single proteins still in a regular microscope, but we need a special, and I will talk a little bit about this, special tricks to be able to see that. But it would be ideal that at one day we can actually see everything without having to do those tricks. So I give you an example now of an image that I took in a super resolution microscope. So this is the bacteria that I show you. I just took a 3D image of the bacteria. So in green is the replication machinery, and in red is the membrane. So that's the membrane, and this is how the cells basically the purple one is the division ring, is where this is a cell that is about to divide. So what is fascinating about this type of microscopy is that you can see things that you otherwise are not able to see in regular microscope. So what about, for example, we're coming back to light microscopy. I already showed you this movie. Why it's also important to have a microscope that allows to see things in real time. So for example, in this case, in this experiment, I will show you, this is just cells growing normally. In this experiment, I said, okay, I want to block division. I want to prevent the bacteria to make new daughter cells. So what happens if we do that? So if we block bacteria, then it just starts to grow like a huge stick, right? But what is interesting is that replication keeps happening. So you have, if you think about this cell, it's a gigantic cell that has a lot of genomes inside, but the genome doesn't know that it's not dividing. It just keeps going. So one of the reasons why I did this experiment is because I'm interested in finding out how does the cell coordinate everything inside, like especially replication with growth. How does, if you are growing, you need to make space for DNA, and DNA has to catch up and fill, the amount of genomes, right? So how does the cell coordinate this process? So I did this other fun experiment where I said, okay, now I block division. Now I want to get rid of the DNA. What happens? So in this case, you will see that the machinery is still working. I delete the DNA, and then you don't see the dots anymore in some of the cells, right? But if you look closely, there are some cells that, here are cells that have no DNA. They don't explode. They're just there sitting. They don't grow. But some cells is still divide, right? So for example, here, there's still some kind of constriction, right? So this is an interesting experiment where I was thinking about, it seems that if you block some of these fundamental processes, the others don't know, right? In bacteria, it's a bit simpler than in mammalian cells. In mammalian cells, this is heavily coordinated. But this is just to show that in bacteria, sometimes the regulation is a bit simpler. In this case, I deleted the DNA. The cells has no DNA, but they still don't die. They just don't grow. But what do you guys think that the cells don't grow?
Because they don't have the instructions in order to make new components.
Yeah, you can think of a simpler answer in terms of like, you need to make ribosomes that make proteins. But to make ribosomes, you need to transcribe from the DNA. And if there is no DNA, then we cannot make ribosomes. So your answer is correct. But yeah, so if we don't have proteins, the cells don't grow, right? So I'm gonna finish now my presentation. I think I'm a little delayed, but I promise that I will be done soon. About viruses, well, we're gonna do some activity with viruses too, with coronavirus. So viruses, you can think of it as the smallest organisms you can think of. But actually that's not correct, because there's also prions, which are even smaller. But people still haven't decided they can call it organisms or not. So why viruses are important? Viruses are predators too. So in this case, it's a virus that kills bacteria, right? And it kills around 20% of the bacterial mass on Earth every day. So viruses are actually very important to maintain the homeostasis of bacterial populations. So you can think of it as I don't know how to say this in English, the food chain, cadena alimenticia, right? So when predator eats the prey, so bacteria in this case are eaten by viruses. But why viruses is so important? Because thanks to them, bacteria develop the immune system, which is the CRISPR. And this has become an extremely useful tool for us in science. So that we have to thank the viruses for their existence. So virus has different shapes. I'm gonna talk to you real quick about coronavirus, because we're gonna do some activity about this. So I show you the spike protein. So this is the spike protein is the one that is, this is the RNA, it's an RNA virus. The spike protein actually is the one in charge of interacting with the cell. So you see here the virus, when it's inhaled, it interacts with some proteins that are called the ACE receptors. And what you see in gray that it's moving, those are sugars. Once it binds to this receptor, then there are other proteins in our cells that basically destroy part of the spike protein, right? So actually we help the virus get in a way, right? And this spike protein is actually very, very interesting, because it has this design that it changes the kind of conformation and inserts into the cell and pulls the membrane, so that it can fuse the membrane of the virus with the membrane of the cell. And once the membrane fuses, then it's over because the genetic material can enter into the cell and be replicated. So this I wanted to show just to have the last organisms that we were going to talk about. And just as a kind of, I'm going to run the last polls just to finalize. Sorry. I don't see it. Oh, okay, no, that's okay. I will run that one later. I thought I had it here. All right, so just as a summary, in terms of size of cells, we can think of, in this case, we talk a little bit about yeast. Yeast is about 4 micrometers. E. coli was around 2 micrometers. One simple trick to kind of think about these scales is, if we are the bacteria, so the humans, a yeast cell is around the size of an elephant. So just so that you have it as a reference. Now, if we think of a human cell with respect to a bacteria, the human cell is around 40, in this case, HeLa cell around 440 micrometers. If we think about that in terms of a bacteria, you can think of it as the human again being the bacteria, and a whale being a human cell. So that's for you guys to have an idea of the scales of size of cells. This is just one movie that I made with cancer cells. These are just dividing, and I have a marker that tells me in which part of the cell cycle they are. So if they are green, they're proliferating. If they are red, means that they are going to divide soon. So that's just to show you a little bit of how they look like. And then the last thing is one little question. Do you guys know what is the smallest cell in the human body? Very good, very good. Very good, very good. Very good, guys. So interesting. So I think the white blood cell won. Sounds like. And the sperm cell as second. So actually, the answer is the sperm cell. So the sperm cell is the smallest cell in the human body, and is followed by the red blood cell, and then the white blood cell, which is the lymphocyte. So you guys were not completely wrong. Actually, it's close to a very small cell. So and then the one that picked the Batman cell, it's almost partially right too, maybe, because Batman is a male, right? So produces sperm cells. All right, so that's it for this lecture, guys.